Well, thanks a lot for having me in this talk series, and thanks a lot for inviting me to participate in the preparation of this really exciting proposal. Both Taher and Mattia and the rest of the team and love that we sat down a little bit and sketched out a few ideas that actually made it into a grant, which is great. It really pushes the limits of my thinking, which is very exciting. It's spooky and exciting at the same time. So it's amazing to be part of this. So I'll be talking about the connectome, and what I mean by that, or the way it is used in human neuroimaging and animal imaging, is a description of the totality of the connections of the brain at a very large scale. So we're not talking about synapse level connections or even specialized neural populations. We're talking about distant brain regions and how they are connected. And the word connectome was coined in 2005 by two people in the imaging field to make the analogy to the genome, for example, or the proteome, which is the totality of the respective field, the respective nodes and connections. And so the connectome really describes the brain at the whole brain level. I'll hopefully close somewhere with the notion of multiplex and why we've chosen that word to describe a new vision that we have about the connectome and how we think about it, how that deviates from what is currently the mainstream in the field. I have a few slides at the, let's see, okay. Oh, let me start with the thanks, so we might not get to it. A lot of the data that I will be presenting have been generously shared with us by others, so we're very grateful for those collaborations. Within the MIV group, the most MIV relevant work that we're doing is involving several investigators here, most notably John Beggs, Karen Dahmen, and Howard Gritton. And so I'll be talking about the slow connectome, which is the state of the art, because most of the investigations in the whole brain connectome are based on MRI, and functional MRI in particular is an inherently slow imaging method. I'll talk about the fast connectome using direct electrophysiological measures, and then I'll talk about how we put this thing together into a multiplex. At the end, I have a few slides of another line of work that I conceptually relate to this. If we get to that, I'd be happy to tell you what our MIV-funded grad student has been up to and how that relates to what we're doing in the lab, how that might be important for mind in vitro. In general, the idea I'm trying to push today is that the connectome organization, or you could think about it as an architecture that's dynamic over time, that connectivity architecture matters for what the tissue is computing. So that's the big message for the mind in vitro context, which is that we probably should think about how we, what kind of control parameters or control input we're using in order to guide certain type of connectivity within the tissue that we want to engineer for it to have certain computational properties. So under that general umbrella, I think connectivity organization, even at the whole connectome level, is relevant for the mind in vitro audience. All right, the slow connectome. So let me start quickly by some introduction, because I know a lot of you probably are not familiar with brain data at this scale. And show you what I mean by the connectome. So we typically record functional MRI data from the brain using usually fMRI, functional MRI. We average the signal within voxels of certain brain regions. Now, different people choose different atlases with different granularities. It can be very granular, it can be very large patches, as in this particular case, and I'm happy to discuss those options. But the activity that we observe in fMRI goes far beyond what we know as evoked activity. Evoked is when I show you a picture, I play a sound, your brain reacts to that. Or if I ask you to calculate arithmetic, imagine something in your brain. If something happens in your brain because of an external demand, that's evoked activity. Everything else is called intrinsic or ongoing brain activity. And the organization of the connectome is inherent to the brain and independent of what is happening in the outside world. And so it's a major architecture that's intrinsic. So if you look at spontaneous or intrinsic activity over time, so the participant is not asked to do anything in particular, you will see that the brain activity between certain pairs of brain regions seems to have a lot of temporal dependencies, whereas other brain regions may have less dependencies. And so this was discovered early on in 1995 already, just 3 years or so after the Bolt signal was discovered. The fMRI signal for non-invasive imaging was discovered. Namely that participants lying still in the MRI scanner, not doing anything in particular, just in a task-free wakeful state, would show coherent activity across all regions of the motor system. So this is primary motor cortex on the left and right side of the brain, supplementary motor cortex, cerebellar areas. And these regions would be co-activated if you would ask someone to actually move a limb, for example. But here the participant is not doing anything whatsoever, and yet these brain regions are spontaneously active and their activity is correlated over time. So they built a cohesive network even in the absence of external motor movement demands. Within a few years, people discovered that, oh, wait, this is not just happening in the motor cortex. The auditory cortex shows correlated spontaneous activity as well. So does the visual cortex. So does the dorsal attention system, and so on and so forth. Higher order networks as well. In this case, they're color-coded. Those regions belonging to the same canonical neurocognitive network are color-coded in the same color. Mm-hmm.
Others may have the same question. So when you say activity, does it mean that they are same time?
So in this case, and 99% of what functional connectome research is based on, I would estimate 99%, the vast majority, is based on the signal of fMRI, which is blood oxygen level dependent. It's not actually measuring neural activity. It's measuring the changes in blood oxygenation as a result of neural activity. And the issue with that is that that oxygenation level dependent signal peaks only 6 seconds after the actual neural activity and takes a total of 12 to 16 seconds to even return back to baseline. And by 16 seconds, I've told you half a chapter of information. How can you understand something like language or any other cognitive process when fMRI signal is that slow? Of course, we've come a long way deconvolving different aspects of the fMRI signal in a way that still allows us to get at pretty rapid processes with fMRI, but there are big limitations on what we can actually measure due to the fact that what is called activity here really is only a proxy. It's this signal here is a change in the relative ratio of oxygenated versus deoxygenated blood, which is visible to the MR system. And that's state of the art for the connectome. And so if you make these calculations, for example, Pearson correlation or some other measurement of temporal dependencies, and you do that for all possible pairs of regions in your parcellation scheme, you get something like a connectivity matrix, which we call the connectome. So this would be a matrix representation of the connectome where some regions are much more connected to each other than others. Spontaneously over time. And this is averaged over the total recording time. So this is how the field started with what we call the static or time averaged connectome. So some of the things we know about the connectome, by the way, before I move on, I don't have to get through all of my slides. I really want this to be relevant to the audience. And I know everybody's at different levels of how they can relate to this work. So interrupt me at any time about anything. Okay. So some of the things we know about the slow connectome as it measured, but I call it slow because it's based on this very slow signal. What we know about it is that it explains variability of cognitive performance within individuals. So this is one example where I'm showing that connectivity matrix now not in anatomical space, but in topological space. So here the colors still correspond to those canonical neurocognitive networks I was talking about. But you can see that in certain brain states, they might be more segregated and clustered, and in other brain states, they might just be all mixed up. So connectivity isn't as sharply separated by these different neurocognitive networks. And so in this particular experiment where we gave participants very faint auditory stimuli and they were asked to report whenever they hear it, a modular brain state right before the stimulus presentation helped them to perceive the sound. If you played the same faint sound, exact same sound, at a moment in time where the connectome happens to be less modular, then they miss that sound. To the degree that we can apply machine learning and I can look at your brain in an MRI scanner, and before I've even shown you the stimulus, I can predict, not amazingly, but above chance, I can predict what you will actually do once I do present the stimulus to you. Right? So I can I can predict your perception into the future using the intrinsic connectome state prior to stimulus presentation. It is also well established that the slow connectome dynamics explain variability of cognitive performance across people. So some people are better at some kind of cognitive processes than others and vice versa, right? Some people might focus really well, others might shift better really well, and so on. And so inter-individual differences are strongly associated with connectome dynamics. And in fact, your connectome, that connectivity, time average connectivity matrix I showed you, is highly specific to you to the degree that machine learning algorithms can find your brain among 1,000 other people, just based on the particular flavor of your connectivity architecture. It's roughly similar. Everybody will have all of these networks, but there will be individual specificity to it. It has also been shown that the dynamics, and by dynamics, I mean how that connectivity matrix changes over time, like this, between different states, how that dynamic is heritable. And the way we assess heritability in human studies is looking at twins often. So these are my twins. It's their birthday today, so I had to have the picture. It's their birthday. They're very excited. This is about 3.5 years ago. They just turned 4. So the way we do twin studies is we take twins like this, which are dizygotic and have very little in common other than happen to have in the same environment. We compare them to twins that actually share the same environment and the same set of genes, right? So monozygotic versus dizygotic twins versus unrelated participants, for example, or other non-twin siblings. And so this is how we tease apart the degree to which genetics is determining the human connectome or this connectivity architecture. Should I go into heritability or should I jump over it? Does anybody here go into the heritability? Because the next slide is about heritability. I can. Sure. All right. Okay. So Sun-Young John in the lab used fMRI, so we're still in the slow connectome, and looked at a hidden Markov model chain of states in the connectome. So you can choose any arbitrary number of states, and there is no right answer. There is a hierarchy of different states that the connectome goes through over time. But you can cut it at a level that is informative. So in this case, we modeled 4 states, and they're slightly different from each other. You might not see all the differences, but they are a little bit different from each other. And the Markov chain shows transitions, right, between these 4 states, and we can quantify the probability of transition between any possible pair of states. We can also quantify the total amount of time spent in each one of these states, which is also specific to individual people. So if we represent the total amount spent in each one of the 4 states for one particular participant in a pie chart, you see that this particular subject has very little occurrence of state 2. Like less than 1%, but a lot of occurrence of state 3, for example. And the thickness of the lines here represent how many transitions happened between any given pair of states. So there's certain state transitions that are more probable than others in this participant. And what's interesting is if you look at the identical twin of this person, genetically identical, right? You can even, without any statistics, you can eyeball the similarities here. Whereas you look at a set of unrelated participants and, well, here we picked a set that was particularly dissimilar, but just to show the variety of personal specificity that you might observe. When we quantify the heritability, I'm not going to go into this graph, but this is it's just a measure of the similarity of those factors that I just showed across monozygotic, dizygotic twins and others. So again, this is monozygotic twins, dizygotic twins that come together as a pair but are different. And so we end up with about 40% of the variability of these transitions and state occurrences explained by genes. So it's highly specific to individuals, and it's quite massively driven by genetics. We also showed separately that these individually specific inheritable phenotypes of connectome state sequences are associated with the cognitive abilities of one person versus another person. You're identical, right? Yeah, identical twins. It would be great to be able to do that. So there's a collaboration we have with Frances Wang and Diane Beck in the psychology department. And Frances has developed a way of probing, ongoing mentation versus mind wandering. Like it's not specifically the content of the ongoing thought, but it is when the person runs off of task and starts to mind wander. And she's developed a way of measuring that over time. It's just really hard because you can interrupt people. That's called the probe method, right? But then you disrupt their type of thinking. Like if you ask them, hey, what are you doing right now, right? And you can do self-reports when the person caught their own mind wandering and those kind of things. It's just methodologically really challenging. We are just starting a new data collection to try and, it's a task where we cover very different cognitive domains like speech, like visual working memory, like motion detection across different sensory modalities. And every trial is drawn from a different kind of task, and the participant doesn't know what's coming next. And there is no break between the trials. So they just have to quickly switch between these things. What we're trying to do is to see whether this chain of connectome states somehow adjusts to those cognitive demands and follows those or not. But those are externally imposed just because of the difficulty of trying to get any measures of your own internal thought process. Yeah, it's exciting to think about how methodologically that could be done. I wouldn't be surprised given that thoughts emerge in part from these states. It doesn't mean these states, it doesn't by all means mean all of this connectivity is related to conscious mentation. It is not. But it results in some ways, it culminates in a conscious thought process, or it affects it at least. And so I'd be surprised if there was not a very strong connection. And if the genetic similarity, the genetically driven similarity in these sequences would also manifest in similar lines of thought. Yeah, if I'm happy to chat with you have ideas of how to measure that. That'd be lovely. So let's see. So the problem, this is the state of the art of fMRI. So we know a lot about the time averaged connectome. We know a lot about these dynamic changes in the connectome. But the problem really is timing with fMRI. So I'm showing here one example of one cognitive process, which is just listening to someone singing this nursery rhyme and understanding the melody, the stress points, the prosodic melody, and the other aspects of speech. So if you filtered the speech, the actual full speech signal for the amplitude modulations of the phonemes, and then the syllables, and then the prosodic stress points, which are these yellow points, you see that they happen in different frequency bands. So this would roughly correspond to the delta electrophysiological frequency band, and then you will have maybe theta through alpha, and then you'll have, upper alpha through low gamma. So this is all happening in the brain at the same time, right, in parallel. And each one of these processes actually requires connectivity between distant neural populations. So we somehow need multiple connectivity schemes at the same time. And then these speeds are, of course, embedded within the full sentence. You are able to remember the syntax context of the sentence as you move towards from the beginning to the end of it. And you're able to remember the semantics. And if you're reading a book, you still remember what the beginning of the paragraph was. And all these slower time scales are also embedded within processing of speech. This, by the way, holds true for all kinds of perceptual processes that are sensory modalities. And so what do we do then if we look at the connectome only at this really, really slow level? We're missing a whole lot of what might be relevant for cognition. And so that brings me to the next part, which is the fast connectome, right? So I put in a method slide here because looking at the entire brain at the same time using electrophysiology, especially if you want to do it non-invasively, only leaves you with a few choices. So you can have electrodes on the skull, that's electroencephalography, or you can have a magnetoencephalography, which uses squids and looks at the magnetic fields associated with the electric activity. But the reason people very, very rarely use this for connectomics is that you have an inverse problem, right? You need to go from what you measure on the skull to where the actual sources in the brain were, and it's a mathematically ill-posed problem. There is no, definitive correct solution. And so people have been kind of really careful and cautious with actually tapping into these real-time methods, even though EEG and MEG allow to actually measure the actual neural activity as opposed to just the oxygenation level dependent signal. That's a proxy, a slow proxy of that neural activity. So the way this unfolds is typically we take the signal as it's measured in every electrode. We might choose to bandpass filter it in these different frequency bands because they're associated with potentially different functions. We don't have to. The electrode level data, so this would be a high density EEG grid, is then source localized to a dense mesh. And then the source signals are averaged across parcels of the brain, similar to the way we average the fMRI signal. So you choose a parcellation that's appropriate for your question and your data. And then for every parcel, you have that average signal. You can analyze that signal in terms of its phase or in terms of the analytic amplitude, so the strength of the signal overall. That's the blue signal here. And then you can, for every pair of brain regions, measure the temporal dependencies either of the phase or of this amplitude. So phase coupling or amplitude coupling of these rapid oscillations of the brain, which allows you to have a connectome like this, right? You can generate a connectome that's similar to the one we have observed in fMRI, even though the signal, of course, is having much, much poorer localizability. Okay, so I hope to show you a little bit of data that convinces you that EEG connectomes are actually not just pure noise, or worse even still, just mathematical illusions, because electricity flows over the skull, we have source leakage. And if you think that region A and B are connected, that might just be because a signal from an entirely different brain region was projected onto both electrodes, and it looks like connectivity to you. And how do you know that that's not the case, right? So there is some mathematical tricks to try and get around that to a sufficient degree. All right, so Sanyang applied the same twin study design now in a very large group of twins from Minnesota that they've been recorded for decades now and followed up since childhood. This is data from their adulthood. And we apply, again, hidden Markov modeling to get different brain states. And what I showed you before for fMRI was 2.5 minutes or so worth of data. Here, we're just looking at 24 seconds. And even within this zoomed in time frame, you see a lot more state switches happening. Much more rapid states here when the connectome is modeled from scalp EEG. We again quantify the transition points and the overall occupancy. We do our statistics. And we show that for different frequency bands, here just two examples, but for different frequency bands of the brain, we actually have 38 to 60, over 60% of variance of these dynamic state shifts explained by genetics. And what that tells me is that the EEG connectome and its time-varying dynamics that we're looking at is actually meaningful and not just noise. To support that notion further, we used canonical correlation analysis to show principal components of these connectome dynamics and their association to cognitive factors. So we find significant associations, without going into details here, between different cognitive control functions and the individually specific inheritable dynamics of the brain. So your particular flavor of connectome dynamics is strongly predictive of your cognitive capabilities, measured separately. So all of the brain data is during task-free rest in this case. It explains the cognitive batteries that have been measured. All right, so I spent quite some time on that just to convince those who haven't heard about EEG-based connectomes that it is possible and that it is in our hands as meaningful as fMRI-derived connectomes are, but it gives us much more temporal resolution, despite of all of the weaknesses that it comes with. So the next thing we do is actually putting everything together. So we take fMRI signals and electrophysiological signals and record them at the same time to then really understand how the brain solves this problem that we need both rapid processes and slow processes to solve any kind of cognitively relevant computation. Okay, so I like to start with invasive electrophysiological data first before I go to the scalp, just because typically the audiences I present this to, even at this point, are usually not convinced about the non-invasive EEG connectome. So I really first want to establish one of our major messages using intracranial electrodes in the human brain, because we are not worried about volume conduction that I just mentioned. We're not worried about artifactual connectivity. What we would see in temporal dependencies between different electrodes would be real neural connectivity or correlation. So this is a piece of the brain of a human where the skull is removed by surgeons. And so this is only done for obviously medical reasons. Some epilepsy patients, unfortunately, do not respond well to medication. And so surgeons sometimes decide to put electrodes under the skull on the cortical surface or directly into the deep tissue. So they go through the cortical layers and even subcortical brain areas and observe the patient for a week or two to just see where the epilepsy is spreading from, and then decide if they should surgically remove the core from which that spread comes. During this time, the patients are in the hospital and typically bored and willing to do experiments. And so we are very thankful for this extremely rare data, because what's special about this intracranial EEG is that it is recorded concurrently with fMRI. So the patients, while they had their electrodes in their brain, were actually put in the MRI scanner. And you might imagine that that can be quite dangerous if you're not really set up for doing that. That's why there is very, very, very few sites. I think I only know of 2 sites who are recording this kind of data. So this is very rare data, and it allows us to answer some important relationships in terms of connectivity organization and fMRI and electrophysiology in the absence of artifacts on the skull. So I first introduced a method here that has been applied just to fMRI a number of years ago, and then I tell you how we take it to a multimodal setting. So what you see here is not a connectivity matrix. Instead, it's spatial correlation of a functional connectivity matrix at time point 1 with the functional connectivity matrix at time point 2. It's a measure of self-similarity over time. So the total recording time in fMRI was 20 minutes. So times is running from top to bottom as well as left to right, and corresponding time points are simply on the diagonal. So what this is asking is, the state of the connectome, that shape of the connectivity matrix I saw at the beginning of the recording, does it ever reoccur later during the recording? If so, we see a high spatial correlation value. So it's really a measure of within recording, within data modality, subsimilarity over time, recurrence of certain connectivity states. What we do with this method is take it to a bimodal setting. Questions up until there? Is this kind of clear? Yeah. Mm-hmm.
But what is the scale at which? So what is the scale, the spatial scale at which fMRI operates? I mean, do you need the big brain like a human brain? Can you do it on a mouse? Can you do it on a engineered
mice
system?
have these networks too, and there's a lot of connectomics work happening in mice. So we have an expert here in the room. If Yazin wants to say more about this, but
and can you go smaller than that?
I
mean, can you do it in a system?
I mean, and I let Yasin go into this some more, but when your recording matrix is smaller and you need less slices to cover the brain, you can go faster because a lot of it is sequential. I'll show a little bit of data that's simultaneous multiband, where you, instead of going purely spatial, recent MR scanners can go a factor of 4, sometimes factor of 8 slices at the same time, and then move to the next. But there is some sequentiality involved. And so if you have a smaller brain, my assumption is you can go faster. People who want to go fast in larger brains often scan just a slice or two, and they want to really establish something at high temporal resolution. The problem with fMRI is, even if the scanner goes faster for smaller brains, or otherwise like tricks like simultaneous multiband, you will not get over the temporal limitation of the bold fMRI signal. No, that's inherent to the biology, right? But the voxel sizes in the typical data sets we use are, right now we're at 1.8 millimeter in plane, a little bit coarser across plane. You can go to 1 millimeter, you will lose temporal resolution for it. But, Lizine will probably have, if I wanted to optimize it, I would go to her and ask, hey, how small can I go?
But sure.
Yeah, so to quickly say something about the small fMRI resting state in small animals, you can do it as far as I know, any mammal, it can be done. The problem is your signal to noise goes with voxel size. So you go to the third power. So that's where you run into problems, is that you lose signal to noise basically for animals much smaller than mice.
Something that can be, mitigated with the scanner, the 9.4 Tesla scanner that gives you higher resolution.
It can be, but field strength goes to the power, the signal to noise increases to the power of 1, linearly slightly above some percent. Then you have cryo quartz, which is superconductive cold quartz, which is doubling signal to noise. So you go high field, cryo quartz, and then you can improve. But that power of the third power is quite hard.
All right, thank you.
Yeah.
So it's correctome assumed to be.
There's people remote. This one makes sure.
Yeah, so these correctomes are assumed to be dependent on a stimulation, or is it intrinsic?
Intrinsic. Most of it is intrinsic. There is quantifications with fMRI data at least, and we've published a quantification. Actually, Parham has done it in the data I'm about to show you. Invasive data, both in electrophysiology and in fMRI, the overall organization of what is strongly connected versus weakly connected stays the same. Even while I'm talking, the rest of the brain still has its own, that's more or less stable architecture. But when I show state differences, when I show that, Markovian approach, for example, and I'll show some other approaches here, there is a subtle change, that's a sort of a flavor, a variation around that attractor that's sort of more or less stable. The external change, how much external demand change it is very little. So that has been quantified. It's quite shocking, actually, to what degree the brain is all internal. And the few slides I have about mine in vitro at the end is emphasizing that the brain is an inference machine. It uses internal states to do computations. It's not an input-output machine in a sense that it always produces the same output for the same input. It's highly dependent on the internal state of the brain, which really is all of this intrinsic ongoing activity. Yeah.
So during development and during learning, the connectome remains the same.
It changes.
Oh,
It
connectome
changes, reflecting both developmental stages as well as aging.
I see.
These major networks will be recognizable. I mean, at very early childhood, they come on slowly, and some of them change a little bit. Well, I don't want to go into detail of some of the changes that have been shown, but it's definitely a reflection of neurodevelopment. There is also finer grain changes. So when people are trained, for example, on a motor sequence task for like 6 weeks, I make you every day, I make you do a certain sequence, right?
Or learning music, playing.
Learning
yeah.
music over a few weeks. It is possible to see the connectivity change between the motor hand area and somatomotor hand area and other relevant regions. So,
Okay.
so the way I think about the connectome is some of it stays the same across mammals as well. I mean, mammals have most of these networks in their own way. I think of it as the history of what makes my brain. That includes my personal history, all of my experiences on all of my learning, be it conscious or unconscious, including implicit motor learning and everything. It's also my history as evolutionary, because those networks have developed and to some degree stay, but then become my personal flavor of it. And so it's I think of it as the total history of my brain. And that history, it's not a vacuum. It is within the intrinsic activity that's relying on the connectivity, the anatomical connectome, which I haven't talked about much at all. It is within that computation happens, right? And so that history affects cognition, even at the level where I said, if I look at your connectome now versus 30 seconds from now, I can predict if I give you the same stimulus twice, how you're going to perceive it this time versus next time, right? So it's to that degree that the immediate history affects perception and the longer history too.
I see.
This is the way I think about it.
Yeah.
Maybe not everybody would think about it this way.
Genetic effects in, that genetic similarity or whatever affects the connectome or the response. And maybe I'm too ignorant, but I don't see exactly the connection. I mean, what you stated in the later part of your talk was more about learn things, about, things that happen historically, right, for the person or whatever, to the brain in discussion, not about similarity from birth or whatever. I mean, there are, of course, a network you inherited, but you can say that you're saying even that with animals, right, we are, kind of behave similarly. So it's all about experiences, if you want, or memory, story, previous experiences.
Yes. So I think it's a combination of all of those. So the genetics wasn't 100% explaining the variance. So
How much percent was it?
so in the fMRI, we found around 40% for the dynamic sequences. Now the static organization is a different beast. I haven't talked about that. So if I average everything over time and look at the heritability of that, that's a different topic. But for the time varying dynamics, what happens from moment to moment, it was about 40% in the fMRI derived connectome and between 40 and 60% in the different EEG frequency bands. And that modeling has a lot of limitations. There's a lot of very approximate assumptions that go into that estimate. So variance component modeling assumes that there is no such thing as gene-environment interaction, which we know for sure is a very strong interaction. It just says there is genes and there is common environment, right? Or non-common environment. So for twins, they have common environment, which is also not true because twins never have the same in utero environment, and even later, they might be treated differently or they might, act differently, and therefore, they're acted upon differently. And so there's a lot of simplifying assumptions. This is really rough estimates, but I think for me, the biggest problem with that model is that there is no gene environment interactions, which is just untrue because identical twins end up having somewhat different cognitive outcomes. They can have different mental disorders. They may have there is a lot of differences that still occur. And so genes can't explain all that. And even if that were fully true, we would explain only 40 to 60% if we accept the simplifications of this model.
Because you see, when they can have the same genes, means they are from the same family or whatever, so they might have actually grown up were developed under the same circumstances, environment, etc. So their similarity comes from there, not from the way they were born. If I would take two twins and put them in, different countries, and they grow up that way with different
Mhm.
parents, that would be the baseline. How much would be similar?
Yeah, so those are hard to come by. There are some data sets that have them. We don't have imaging data, not in a large scale. So heritability requires a quite good number of people to have good statistics. Like you don't want to do it in small sample sizes. So different environment, twins growing up in different environments are much more rare. But if you look at monozygotic twins versus dizygotic twins, with the simplifying assumption that both types of pairs of twins share about the same amount of common environment, but the monozygotic share on top of the common environment, the common genes, right? Then that's kind of the model that we're using. And it has to be a combination of all of these. Where else would learning go if it is not reflected in the connectivity of the brain? Right? The question is it reflected in the connectivity brain at this coarse scale we're looking at? And I'm thinking it is.
Well, you mentioned that there is something like expressed some other way, storage, memory. I mean, it's not only input-output machine.
Yeah, but the memory of the brain
state.
is its full connectivity organization, right? And the full connectivity organization is a world larger than what I'm looking at, which is a very simplifying, averaging over large patches of the brain to make a coarse connectome. I mean, there's such things as voxel-wise connectomes, but they've not gone very far. It isn't very useful to think about it at that granularity. But the question is not whether memory is in the connectivity of the brain. That is I'd be surprised if that would be disputed. But the question is it reflected also in the connectivity at this coarse whole brain scale, or is it just in the fine synaptic connectivity that I can't have access to with these kind of methods? Is that,
Well,
or do you think there might be other ways of storing memory than the connectivity? Right, but the chemicals are released by neurons, right, or other cells that are, and what signals that they should be released in one way or another is driven by their connectivity again, or who they're influencing is depending on where they're innervating other areas, right? So well, my lab is called the Connect Lab. I put all my eggs in this one basket. I'd have a hard time. That I reduce the chemicals to the connectivity. No, there is a strong interaction between. In fact, I've taken the slides out of this because we're in the third revision of a submission and a reviewer is grilling us about it, that I'm starting to feel not as confident about mentioning it. But within the same twin set where I showed the fMRI Markovian chain states, not the EEG, in the fMRI, we actually have the full genome of those participants, or we impute whatever is missing. We're showing that certain particular polymorphisms in the chemical neurotransmitter systems are related to that particular individually specific and inherited type of connectome dynamics. So we show actually a relationship to the genetic polymorphism in the neurochemical systems. So I'm all about the neurochemistry driving these connectome state changes. So if you think about the ascending neurochemical pathways, because they are very diffusely affecting the brain as opposed to a one-to-one connection. So not neurochemicals that are synaptic like glutamate and GABA, but the neuromodulatory transmitters that affect large populations at one time, right? Those ascending systems, we've looked at the genetics and those, and they explain a lot of this. So neurochemistry is definitely a big part of the modulatory changes of this connectivity activation over time, because we're not forced into the anatomical connectivity. Otherwise, there would be no connectome states. It would be just that one matrix all the time, right? And we show that that's not fully explaining the connectome. The way the connectivity changes slightly, just slightly around that stable architecture. might be very well driven by these neurochemicals, right? And going through these dynamic states of the connectome. Yep. Okay, so let me tell you where I'm at. So we have a lot of bimodal data here. I could show you not the data and just show you the message, or I can go to Mind and Vitro.
It's up to you. It says here, meaning that there's some restlessness.
Would you want people to grab pizza and continue the conversation, or?
You can, I would say, if you can jump to the conclusion, that would be useful.
Great. Okay. So all right. Okay. So the way we analyze bimodal data, again, I said we have the intracranial bimodal with fMRI, and then we have also scalp like this. So we take concurrently recorded fMRI and EEG. We do the source reconstruction that I explained earlier. We average fMRI signal across the same coarse atlas. And coarse, the reason for coarseness is that EEG cannot localize better than that. If we do a time varying, we need enough SNR for a time varying connectome approach. So at every time point, we might generate this kind of a connectome and then look at the relationship of this connectome over time. We've done that both for the invasive data as well as the scalp data. And the bottom line, and I'm happy to come back to how I got there, but let me show you the conclusion. So the bottom line is that what we think is happening in the brain is that at one given moment in time, we don't have such a thing as one connectome state. So it could be that at a relatively rapid state, at a relatively rapid time scale, if we filter the EEG data in this really rapid time scale, we might find connectivity that is in a particular brain state. Certain regions are highly connected, others are less connected. But then at the same physical moment in time, in the delta band, there might be connectivity across a different set of brain regions. And then the infra-slow time scale that fMRI images so well and EEG not so much, we find yet another connectome state. And so that's what our bimodal data is suggesting. And the reason we like to think about it as a multiplex is that, you know, at every moment in time, you have a stack of connectivity. And if you think about multiplexing and telecommunication systems, you'll have one single system within which you can tap into different frequency bands to transmit multiple types of information without interference concurrently. And so that's why we like this idea of a multiplex. It seems that people can more intuitively connect to why we think about the connectome dynamics as a stack as opposed to a single stream dynamic. So the reason most people intuitively would think about the connectome as a single stream of connectivity states is that they're only looking at the infra-slow time scale, because the rest of it has all these methodological limitations that I mentioned, and we're trying to push the field to try and accept and open their mind to more real-time methods concurrently. Right? I know. You should be my reviewer. I mean, until you have concurrent data, you're not going to think about it this way, right? You have to understand that for about 20 years, we've looked at the connectome just with fMRI. When I say connectome, I mean the large-scale whole brain connectome. There's people that work at much finer grained scales, and then that has an entirely different type of meaning. But so this is why it's surprising. Let me show you a kind of a quick.
Mics on that way, people can just talk, but then be careful because you pick up everything.
Right. So the reason people are surprised, at least in the human and large mammal field, is that we intuitively think that EEG and fMRI, or any real-time method, this can be intracranial EEG, this can be MEG, and fMRI measure more or less the same neural processes, except that fMRI forces it through this hemodynamic response function filter. It forces it through this particular function that we have shaped. The shape has now been mapped for many decades. And makes it smooth over time. It's like it's a low pass, it's a temporal low pass filter. But underlying it is the same neural activity, right? And if there is any non-overlap, it's mostly noise and other things that maybe fMRI cannot measure some of the fast dynamics, right? But what it does measure should all be visible in EEG. And whatever their discordance is just limitations of methods. What we're saying is no, there's quite a proportion that's non-overlapping, but that's by biological design. It is because fMRI is measuring the infraslope part of brain activity that's very important. And I'm coming to the line in vitro slides there to show how important the slow history of an agent is over time. That signal is measuring. I think it's a lucky coincidence that fMRI can only see slow things, because we would never have looked at it otherwise. We would have ignored it. I'll come back to the slow history in a moment. But fMRI is particularly good at the autocorrelation over time of the agent, of its recent past and long past. Whereas the EEG is much more rapid and measures biologically different processes. And that's where people are surprised. When I tell them, I'm not saying fMRI doesn't have an electrophysiological basis. I mean, you could close my lab if I said that, because then I can stop my work and fMRI has no meaning anymore, right? That is not the case, right? But there is a big part that fMRI measures that EEG cannot measure very well. There was theories for why that is. I'm not going to go into those, but it's evident that EEG is measuring a lot of rapid things that fMRI doesn't see really well. I'm just saying they are multiple parallel neural processes that we should all keep in mind. And that makes a new, different kind of concept of the connectome. If you only look at this range, you don't even conceptualize. Maybe it is intuitive. It's just that people just don't look at it. So there is no such concept as a stack or streams, a multi-parallel stream of connectome dynamics. Is that?
Yeah, no, I'm just surprised that it's controversial, because in biology, having a hierarchy of spatial temporal scales is a lot of difference.
Is obvious.
So
yes.
Why wouldn't it be
Yes,
this?
so I should say, it is perhaps surprising at the level of the whole brain connectome. If I tell someone the alpha EG frequency band is not just in the occipital cortex, if you look at it through a connectivity lens, you will find entire connectome matrices at the whole, like all by all region scale, that have a small to moderate level correlation with those in other frequency bands and fMRI and structural connectivity. That is what's surprising. People don't think about all of these rhythms playing a role in large scale communication. I think that's another level of disconnect. They would of course accept that if you look within a region at different kinds of cortical layers or within different specialized populations or for different cognitive or cognitively relevant neural processes, you would find different rhythms. That's obvious, right? But that the connectome at the large scale would be associated with all of these EEG rhythms, that is not the way EEG colleagues think about it.
I mean, is it one way to think about? I mean, for engineers, I think it's very easy to understand. So, is it the confusion that when you're looking at high frequencies, a set of neurons in those connectivity map are activated? They're firing at the same time. But when they're at a lower frequency, maybe in the same region, a different set of neurons are activated. There could be two neurons next to each other in a 10 micron space, right?
Absolutely.
So it's like multiple circuits,
Yes.
but in a next to each other, occupying a region.
Yes.
So
And when we average over the region, we
Right.
don't have the resolution to see that. That is one obvious way. Another way would be layer specificity, like cortical layers.
right.
So we have, I remember speaking about this, but we have a layer expert here in the room, but these oscillations are not distributed the same way over the different cortical layers. The simplest way of thinking about that stack is literally the reason we have developed over evolution 6 layers in our cortex, right? I mean, that should make it really easy for biology. I don't know if they map that way. I don't mean them that way. I mean them computationally.
Sure.
But you put one architecture that could afford that computation with either side-by-side populations or a stack.
Yeah.
The other thing that we know of is cross-frequency coupling. So the same neurons we know that, for example, neurons firing in the gamma range might silence themselves sometimes and go up and down